we have to follow some standard rules while we are in the session. So this session will last for 120 minutes. The first 90 minutes will be addressed by the panelists with their presentations and followed by a 30-minute Q&A session. The participants' audios will be disabled during the first 90 minutes of the session. You may use chat feature to share your questions and comments during this time. Keep your videos off when the speakers are giving the presentation. Please use the raise the hand feature at the bottom of the screen during the Q&A session and wait for the moderator to address your name. Well, I think hate speech of any kind will not be tolerated during the session. Making such comments will be removed from the session. The discussion will be recorded and later will be uploaded in given YouTube channel. So these are some of the rules that we have to follow. Well, moving ahead, as you know, digital sequence information is a revolution in science and technology that was unthinkable 20 years ago, and it's still unknown to the public, but the impact couldn't be bigger. To understand DSI, we have to dive into the principle of life on Earth. Every animal, plant, and microorganisms, every cell of the body carries a chromosome, and every chromosome carries DNA. This DNA carries an extremely long string of 4 different chemicals called adenine, guanine, cytosine, and thymine. These chemicals are arranged into a genetic code that determines how an organism looks, grows, and lives. Ladies and gentlemen, DNA sequencing determines the order of the 4 chemical building blocks called bases that makes up the DNA molecule. No doubt that DNA plays a fundamental role in environmental and biological research, contributing to understanding of the molecular basis of life and evolution, and of the ways in which genes can potentially be manipulated to provide new therapies and cures for diseases. DSI has a wide range of applications like producing vaccines, drugs, identification of animals and plants, comparing the genome sequence of different types of animals and organisms, such as chimpanzees and yeast, can provide insights into the biology of development and evolution. But for any of this to happen, first we need to sequence. What does sequencing mean? I think our panelists will tell more about it. Now, anyhow, the sequence tells the scientist to the kind of genetic information that is carried in a particular DNA segment. Now, DSI and the corresponding subjects are being discussed under the instruments of CBDA and various other international protocols. I welcome all of you to this wonderful session called DSI, and hopefully all of us can learn something. It's a very free-flowing session, knowledge sharing session, learning session. So I request all of you to interact in this session and participate vibrantly so that we all can understand and learn from each other. On that note, I first welcome Mr. Christian Squazer from Germany, who is the co-founder of Global Youth Biodiversity Network, to give the opening remarks.
Yeah, thank you so much, Bharat, for this opportunity. Welcome, everybody, to our webinar on digital sequence information. As Bharat said, my name is Christian. I'm from Germany, and I'm the co-founder of the Global Youth Biodiversity Network. And we have the pleasure to host this webinar today together with our colleagues from the Third World Network. And I very much hope that this session will be informative for you, that you will learn something about why digital sequence information is such a hotly debated issue in the Convention on Biological Diversity. I was asked to say a little bit about our network, so allow me to share my screen. As you can see here, this is the extent of the Global Youth Biodiversity Network. We are the youth constituency in the Convention on Biological Diversity. We always felt that just bringing young people together in an international convention and giving them a voice in these negotiations is not enough. We feel that in order to take action for biodiversity, all of us can do something every day in our backyards, in our community, with our families, with our friends, with our schools, with our politicians, with our local governments. And because of that, we're also putting a lot of focus on mobilizing young people all over the world on biodiversity issues in general. Over here on this slide, you can see the size of our network. We have more than 600 member organizations that are either already active on biodiversity or that wish to get active on biodiversity issues. And we have 50 national and regional chapters all over the world. National and regional chapters are groups of young people or organizations that have joined forces on the national or regional level in order to collaborate and take actions on biodiversity. They're organizing all kinds of activities. It can be campaigns, it can be educational initiatives, whatever is appropriate in the context of the country. We also have individual members in 172 countries, and all in all, we're currently representing around 1.2 million youth all over the world. So this is it about Gibbon. So far, we have been mostly active on the so-called classical conservation issues, like protected areas. We have been active on issues related to equity. We have been active to all issues related to the CBD, but we have not been super active on all issues related to synthetic biology, digital sequence information, and everything in general on biotechnology. But we're recognizing that this is an issue that is becoming increasingly important in the Convention on Biological Diversity. And because of that, we wanted to organize this session together with Third World Network today in the hopes that this can build some capacity in our community to better address these issues that will certainly become more and more important in the coming years. So thank you very much for this opportunity, and I hope it will be an interesting event today. Over to you, Bharat.
Thank you for that opening remarks, Christian. We appreciate your time and all the inspiration that you have given to all the youth members. Thank you so much. Now I request Li Ching Lim from Third World Network to give her opening remarks on DSI. Over to you, Ching.
Thank you so much, Bharat, and good morning, good afternoon, good evening to all of you. My name is Ching. I'm from Malaysia, and I work with the Third World Network. TWN is very honored to be co-hosting this webinar with the Global Youth Biodiversity Network. Much appreciation, of course, to the great team who have been working behind the scenes, also to our interpreters for organizing everything. We, TWN, we are an international research and advocacy NGO based in Malaysia, and we've been monitoring the discussions at the Convention on Biological Diversity, or the CBD as it's known, since it was established as a real convention in 1992. Now, I know some of you were not even born then, but my organization has been tracking the negotiations under this international legally binding treaty. Now, the objectives of the CBD are threefold. They are the conservation of biological diversity, the sustainable use of its components, and the fair and equitable sharing of the benefits arising out of the utilization of genetic resources. Historically, developed countries have focused more on the first two objectives, conservation and sustainable use, while developing countries have championed the latter objectives because of North-South inequities. Much of the world's biological resources are located in our developing countries, but the commercialization for profit has mainly been driven by entities in developed countries. As such, the issue of benefit sharing is critical, not only for the developing countries that hold the wealth of these resources, but particularly so for indigenous peoples and local communities, smallholder farmers, who are the ones who are actually nurturing and safeguarding biodiversity. For when researchers or companies use the genetic resources and the associated traditional knowledge, make profits, but don't share the benefits, and sometimes even earn, through patent protection, earn monopoly profits. This is actually biopiracy. It is theft, pure and simple. And now today, with the technological advances in genome sequencing, there is no longer a need for companies to access the actual biological samples. Instead, they can just use the gene sequences, or what we call digital sequence information, or DSI. So the issue remains important for biodiverse developing countries because it is their species that are being sequenced and then placed in unregulated databases controlled by the rich countries. Countries that to date do not expressly acknowledge the obligation to equitably share benefits for DSI use. Now, if users of biodiversity, particularly commercial entities, access biodiversity in the form of digital sequence information, yet don't share the benefits, then the third objective of the CBD is not fulfilled. Likewise, the Nagoya Protocol on Access and Benefit Sharing, which is a protocol to the CBD and is meant to operationalize the third objective, will be in jeopardy. This is therefore a situation of digital biopiracy and needs to be rectified. To not share benefits for the use of DSI would be unfair and inequitable, as the people who have taken care of biodiversity deserve to receive a fair share of any benefit that is derived from the use of that biodiversity. And this principle has to apply, whether biodiversity is accessed in a physical or digital format. As mentioned, discussions are ongoing at the CBD to try to find agreement that would require DSI users to commit to share benefits and to work out how to do so fairly and equitably. TWN and the Global Youth Biodiversity Network, we are therefore holding this webinar to enable our constituencies to understand this issue better. So we're going to have some very introductory presentations because we are aiming it at people who are new to DSI or who have questions about it, and really just an opportunity to learn more and to share and to exchange knowledge and understanding on this issue. Because it's important, because DSI relates to fairness and equity for developing countries and especially for farmers and indigenous peoples. And our goal and our hope is that after today, people will come away empowered and therefore then to be able to advocate for a solution that brings fair and equitable benefit sharing for the peoples who are the ones who are conserving and sustainably using biodiversity. And therefore you see the interaction of the three objectives of the CBD here. And I hope that we have good discussions, and I'm really looking forward to them. And thank you very much. And back to you, Barath. Thank you.
Thank you so much, Ching, for those opening remarks. Incredible. Thank you also for collaborating with us and encouraging and believing in the youth that we can do this together. Now, I would like to invite a very, very interesting speaker, panelist, uh, all the way from United States of America, I believe, Mr. Edward Hammond. Mr. Edward Hammond directs Prickly Research and is an advisor to Third World Network. Mr. Hammond have been working in access and benefit sharing from since 1990s and has extensively written on biopiracy and digital sequence information. And he's member of the CBD ad hoc technical expert group on DSI and served on the ITP GRFA ad hoc open-ended working group to enhance the functioning of the multilateral system. Mr. Hammond, we'll be looking forward to listening to you. Over to you now.
Thank you, Bharat. Thank you for the introduction. I'm very happy to be here. Maybe when Ching said that that GWN and some of us have been around since the early 90s, you didn't believe her, but with my head of gray hair, maybe you believe her now. So we've done a number of webinars, and I've very closely followed this issue since the beginning, which was really at the Biodiversity Convention meeting in Cancun several years ago, when it came on to the sort of digital sequence information came fully onto the international agenda. And in the course of that, many times we've had sort of very intense policy discussions about, you know, what's going on right now in the CBD, et cetera, et cetera, et cetera. But in the course of doing those types of webinars and presentations, many times, you know, people came to us and said, hey, you know, I'm really interested in this issue. It sounds, you know, very important in terms of fairness and equity, but it's rather technical. And it's hard for me to, you know, understand where things are because there's a bit of missing background. So that's what this webinar is intended to do, to provide sort of more background on the DSI issue, and to hopefully have you leave at the end of the webinar feeling as if you understand its basics and hopefully motivated to continue to work on it and be entitled to and express your opinion about it. I'll warn you that my presentation doesn't give you any answers. And the reason why is that it's really about presenting the problem. I will be followed by another presenter, which will go on and will describe more. This is intended to be the first in a pair of webinars. So later this year, we'll hopefully have a second webinar, which will go into the questions of the immediate politics and what's happening in the CBD today, and what the solutions are. And certainly we can talk about solutions some in the question and answer at the end of this session. But for now, for this presentation, the focus is really on what are the basics of the digital sequence information issue, why has sequencing become so important, and what are the implications for the major groups that are interested in biodiversity and its conservation. So I will share my screen now and go through the presentation.
Edward,
yes.
just before you give your presentation, I kindly request to reduce the pace of your speech for the interpretation. I'm sorry for that, but
okay, very good. That's fine. All right. So, excuse me. Let me just move ahead here. All right. So there are 4 major bits of the presentation that I'm going to give. The first is understanding how gene sequencing is changing biology and really has already changed biology fundamentally. The second is to discuss the applications for digital sequence information. the ways in which it's used. The third part of the presentation is, well, what is the scope of digital sequence information? Is it just gene sequences, or is it more? And then finally, we'll start to talk a little bit about the policy and political issues a bit more. And that's, what are the impacts on farmers, on the CBD, on developing countries, and the various constituencies that are interested. So we'll start about talking about sequencing technology. The CBD was completed and was initially signed in 1992. That was 3 years before the first organism was actually fully sequenced. That organism was simply a small bacteria in 1995. And a year later, a somewhat more sophisticated organism, a yeast, was fully sequenced. These organisms that were sequenced now more than, 25 years ago almost, were organisms that had between 1,600 and 6,300 genes. So the number of letters in the alphabet of these organisms was a little bit less than 2 million in the case of bacteria, in that particular bacteria, and around 12 million in the case of brewer's yeast, which is what or baking yeast, which was the other organism that was first sequenced. Following on that, the health authorities in the US and in other countries decided to sequence the human genome. And using the technology that was available to them beginning in the 1990s, the project was shared over 20 labs, an international collaboration of 20 labs. And those labs worked for 13 years, from 1990 until 2003, to create one sequence of one human individual in draft form, really, in complete form, but not in what would be considered a high quality form today. It took that project, which ran all through the 90s and into the beginning of the 2000s, 13 years. And it cost, in today's dollar terms, $5 billion. So this is an enormously complicated, slow, and expensive process in the 1990s as the Convention on Biological Diversity is being implemented. At that time, it took this sort of investment to sequence at a meaningful level. So the key points in the sort of old sequencing technology, what we're going to contrast against the situation today, is that back then you might hear, well, we have sequenced the sheep genome. And what that would mean, or we've sequenced, say, the bean genome, field beans. What that would mean would be one individual, one particular representative individual out of the entire population was selected and a sequence created from that. So what should be obviously apparent when I say that is that, of course, people and all species almost, there's some species which are rather uniform, but most species are incredibly diverse. And so when before, and 20 years ago, even 15 years ago, when we talked about sequencing the genome of a human, we were talking about sequencing the genome of one person. And that's a very, very different thing than sequence information that represents the full diversity of humanity, or elephants, or plants, or whatever organism you're interested in, than the sequences of everything. So fast forward only 20 years, really, less than 20 years, really, to where we are today. Whereas in the 1990s and early 2000s, it took $5 billion and 13 years to do one human genome. In a few days and $300, you can do your own genome today. This is the box that's in the upper left of the slide. The particular company whose image that comes from also wants your data. If you want a modicum of privacy, you probably pay about $600. But it's become a routine and relatively inexpensive thing, even for very high complex organisms like ourselves. And the cost has obviously come down tremendously. Instead of $5 billion to sequence a human genome, it can be done, let's call it $400 or $500 as being the cost now. So it's much less expensive than it used to be. Previously in the 90s and in the 2000s, if you look at the lower left of the slide, sequencing took place in enormous rooms with enormous machines, one stacked right after one another, each of them costing 60, 70, 80, 100, 200,000 US dollars, working full time to do what can be done now with a USB stick that costs less than $1,000 in about a day. So in addition to becoming incredibly cheaper, the technology has become incredibly more compact and transportable. And in that time, as a result, if you move to the upper right part of the slide, we've gone from this conceptualization of what it means to sequence a species has changed from sequencing one representative individual of a species to sequencing incredible numbers. So now in agriculture, for example, there are projects to sequence not one field bean variety, but to sequence 30, 40, 50,000 of them. And that creates an enormous amount of data, and we'll talk in a little bit about what the implications are of that. And now, particularly driven by the COVID pandemic, this whole notion of genomic surveillance is becoming rather prevalent, and people are beginning to talk very seriously about sequencing all samples of a species of interest globally or within a country or a region and sucking up all of that data. And there's an effort in effect to do that with COVID right now. Now, COVID is a very small organism compared to a human or a mammal or a plant. Having said that, if you look how far we've come in 20 years, a reality in which we can sequence every member of a national population or every individual plant of a particular species that we can find, maybe something closer to a reality. And the slide shows just an effort. Those are COVID strains from different regions of Denmark. And you can see how deep the sequencing goes, that many strains from every region are being sequenced and are being organized in a taxonomy. So, and that leads to the bottom, is that now you have proposals out floating and people working on the idea of, in addition to sequencing of all the members of species, but there's also sequencing is moving across biology, and you have things like the Earth Genome Project, which want to sequence at least one and hopefully a lot more of every species on the planet. So this is really becoming ubiquitous. That is the state of the technology now, and there's little reason to suspect that it's really going to slow down anytime soon. In fact, the commercial interests and to an extent academic interests for different purposes are accumulating data as quickly as they can, accumulating data, in fact, more quickly than they can figure out how to use it. And that is actually points to one of the issues in the politics of this, which is there's a bit of a rush going on to establish collections of sequences. So where do all these sequences go? So you have, lots of guys carrying around USB sticks who are having computers in rooms and equipment, and they're sequencing everything. Where do they go? Well, some of them aren't particularly useful and stay in the lab. Some of them are hoarded because they are seen as potentially valuable by whoever sequences them, and they keep them private. But many of them, and those that are published in academic studies or in anything that's studied, whether anything that's published, be it academic or not, go into what are called open access databases. And the 800-pound gorilla, or the 350-kilo gorilla of open access databases, is called the INSDC, which is the International Nucleotide Sequence Database Consortium. And this is a set of 3 parallel international databases, which are enormous, which function as a sort of triumvirate of Japan, Europe, and the United States, support each of the databases. But they sort of pretend that there are 3 different databases, but they copy each other every night. So they're in effect 3 copies of the same database. Some of the tools that are available at the different portals where you can access these databases may be different, but effectively they are a single database. And in terms of policy and in terms of understanding where the publicly available sequence information is, INSDC is critically important, because they have all the information that is open access, or a large proportion of it. And also other open access databases tend to work with them, and they copy back and forth some. So their policies and the way that they manage all this information is really central to the issue. Now, we've discussed this in agriculture for gosh, 10 or 12 years now. But it's also relevant in other forms of biodiversity, other types of biodiversity. There are also corporations that we know are building enormous databases. In fact, although they very seldom want to admit it, the INSDC serves more data to corporations who download the entire database by FTP than they do over their web portals. So we know, but no one knows the details of, except the privileged people within corporations, of what's going on with the private genomic databases. But there's every reason to expect that they're huge, because they include everything that's in the public, or in the so-called public domain, or everything that's in open access databases, plus anything that these companies view as being relevant to their business that they've also acquired. So we know with certainty that the big biochemistry companies, be it in agriculture, be it in health, or even companies, be it that are in the business of sequencing and interpreting sequences themselves, are all amassing enormous private databases, and they're trying to leverage this data with artificial intelligence to try to guide their product development. And I won't go into this in great detail, because our next presenter is going to say a lot more about the big data issues. So we've talked about where the sequence information comes from and how that technology has evolved. We've talked about where it goes into these databases, public, private, open access. And now, so what are the applications? How is it used? I mean, the first thing that you have to say is that there are some uses of digital sequence information which are relatively uncontroversial and which most people find harmless or even helpful. So it can be used in taxonomy, when you have, like in the image there, when you have a lot of, when you have species that interbreed or that are very closely phenotypically related, you can't tell the difference between them. Sometimes running sequences will help you sort them out. There's also use of digital sequence information in trying to enforce endangered species law. So trying to prevent the entry of endangered or otherwise, prohibited species into either the industrial or the food chain. And finally, there's use of DSI in food safety, trying to prevent, obviously no one wants to get ill. So those are just three examples of applications of digital sequence information that are generally regarded as being beneficial or useful. And then there are others which may be beneficial and useful as well, but which raise a lot more questions about ethics and fairness and equity above all. And that would be the use of digital sequence information. And the two major areas that I'm going to talk about are in proprietary medicines and in seeds and agriculture. In medicines, I'm including vaccines. There is a very practical and advanced use of digital sequence information today. A lot of viruses, particularly RNA viruses, those are viruses whose code is composed of RNA as opposed to DNA, can be sequenced, converted into an email, sent around the world, and recreated in a matter of perhaps 48 hours now. So we've crossed the threshold of being able to do what might have seemed like science fiction when I was a kid, and that's to sort of beam an organism from one place to another without physically carrying it. The sequence information that's generated, particularly from pathogens, is an interest of ours and an interest of mine, because it's used to develop the therapies. And the big one that we're all very conscious of right now are COVID vaccines. COVID vaccines, effectively all of them, some of them in more direct ways, are products of digital sequence information. They would be impossible to have now and to produce were it not for the availability of gene sequences of COVID strains. And not any COVID strain, but there are particular COVID strains when the organism changes that are of particularly high value. So this is without getting too much into the politics of this, this is an important issue when you think about the problems that most of the world is having right now with accessing COVID vaccines at all, or accessing affordable COVID vaccines. Well, at the same time, scientists the world over, including developing countries, are being encouraged, cajoled, pressured to continue to upload these sequences because they feed the continued development of vaccines, but those vaccines are only available to some people. A concrete example of this would be, it's not a vaccine, it's a monoclonal antibody. It's a type of treatment for Ebola. There's only one approved treatment specifically for Ebola. That was developed by a US company. The US company didn't go to Africa, doesn't have any arrangements to where Ebola comes from, doesn't have any arrangements to share benefits with anyone in Africa, because rather than obtaining a sample and signing an agreement, they simply downloaded the sequence from GenBank, from an open access database, developed their product using that, their patented product, using that quote unquote open access information. And the company has received at least $1 billion in US government funding. And we have a situation now where the United States government is paying hundreds of millions of dollars to stockpile tens of thousands of doses of treatments for Ebola, but there is no availability in Africa except for the occasional donation of relatively small amounts from the company. So that's a a short picture of what is happening or how the issue applies in pharmaceuticals. But it also applies in agriculture. And one thing I want to flag here is this also shows the different ways, right? In the case of the pharmaceuticals, at least the ones that I mentioned, it was individual sequences of individual organisms that proved to be very valuable. And that's the case across the board. It can be the case in agriculture, but really in agriculture, the major focus is in aggregate use of DSI. So if you are, for example, looking for a trait for a plant that's going to make it more tolerant of drought because you're selling seeds in a region that's becoming drier because of climate change, the way that you might be able to do that, and enabled by digital sequence information, is to look at how many different individuals and varieties of this plant population perform under different circumstances in terms of the amount of water available to them. And you may identify genetic patterns which may be quite complicated, not just a single gene change, though it could be that, but you might identify through the course of looking at massive amounts of data associated with information on where and when and how much rain occurred that year, and be able to guide a breeding process to create a new variety of plant. And this is being done on a proprietary basis already by the major international companies. And then there's a whole sort of ecosystem of companies related to that are providing analytics, providing artificial intelligence, et cetera. So this is another key area because where do the seeds come from, or where do these varieties come from that are being analyzed? Well, in many cases, they're farmers' varieties, which have greater diversity and which have been grown over a variety of circumstances over many years. You're not going to learn quite so much if you're looking at 50,000 sequences of the same Monsanto corn or maize as you are if you're going to look at the diversity of environmental response of a huge variety of farmers' varieties of maize. So that's a bit about how it plays out in the world of agriculture. And of course, these are only two applications of DSI. There are many more. I'm running a little bit behind here. So this is the only complicated slide that I'm going to show, and you don't have to pay attention so much to what the detail is of what's in the box that you see right now. But what this is a way, and I find this box actually pretty useful, is a way to talk about what is the scope of DSI. Because when you put an organism into the machine, well, machines are getting more complex, but as a sort of generalized statement, the machine is just going to read the letters of the DNA. It's not going to tell you what those letters mean and what that sequence of letters does. So in the policy and the political debate over DSI right now, there are a lot of questions about, well, what does it include? Does it just include those raw letters, or does it include other things? And I think, of course, it does include other things, but the question is, how far do we include other things under digital sequence information? And that's what this table does. And if you can see that I've outlined some in red, the red box at the bottom, which are things that I would say are not digital sequence information, and the green box in the top. So what is included in the concept of digital sequence information, at least for civil society organizations, and I think most indigenous people and local communities that I know of that are working on this issue, is, of course, those letters of the DNA alphabet, but also the information about how to put it together, how to organize those letters so in their correct order, and how to break down those letters into meaningful segments. Like this segment is a gene that has this function. So also information about the functions. And finally, there are additional bits, heritable parts of how the genome is structured. And then the letters in the alphabet encode amino acids, which in turn create the molecules that sort of power cells and keep us alive. And so the molecules that are directly encoded are also generally considered to be part of digital sequence information from our perspective. This is a subject that is going to be of considerable debate in the sort of coming years in the Biodiversity Convention in all likelihood. What is not digital sequence information, in my view, is the type of knowledge that indigenous people and local communities, that farmers have about these crops. Things about knowledge about how particular crops perform under certain conditions, knowledge about medicinal or culturally significant uses of crops. These are things that fall outside of what I think is properly considered DSI. So when we're talking about DSI, to make it short and sweet, you're talking about the gene sequence reads, how they're organized, and the immediate molecules and bits of molecules that they encode, and not the sort of human knowledge of the type that farmers and indigenous people have about them. So now in the final bit of the presentation, just to talk for a moment about what the implications are. And I already warned you that I don't have the solutions here. If you want the solutions, we're going to have another webinar. But what's happening right now is that the traditional world of bioprospecting and the traditional way to access organisms was that a researcher, and that's depicted in the top of this slide, is that a researcher comes to a country, goes to farmers or indigenous people, says, wow, I'm really interested in your plants or in your medicines or whatever it is. I want to collect samples and go back and study them in the lab. And the Biodiversity Convention set up systems to deal with this. And under national legislation, those bioprospectors would have to sign an agreement, and the agreement, a material transfer agreement. And this physical exchange of materials created legal obligations. And those legal obligations, at least in theory, made it compulsory for commercial users later on. So the bioprospector or his friends at the corporation that he gave it to, or maybe the bioprospector patented, a medicinal compound and sold it to a company. So in theory, this national legislation and physical exchange of materials would generate benefits that went back to the country of origin. Well, that model is becoming obsolescent. And the reason why the model is becoming obsolescent is because of data, is that things are being increasingly sequenced in place. And those sequences can be used in lieu of making all of these commitments under the material transfer agreements, and the transfers of the relevant, useful parts and information about organisms can be made digitally. So this is a tremendous challenge to all of the systems that have been set up since the 1990s in the Biodiversity Convention, because the benefit sharing, the third objective of the Biodiversity Convention, is fair and equitable sharing of benefits. And we had a legal means by which this was done. It was national access legislation and, material transfer agreements. Well, now that's being challenged. Now, some countries say that their national access legislation will apply to digital access. In practice, that's pretty tough to do right now. So this is one of the issues that's being confronted. So the last slide really is this, to say, so what are the challenges of the new world of sequence information? You know, for the Biodiversity Convention, it's really an existential threat. For the Biodiversity Convention rests on 3 pillars. The 3rd pillar is fair and equitable benefit sharing. If you have massive theft of all of the uniqueness, all of the diversity that's coming out of particularly sort of the really super diverse countries, most of which are in the developing world, if you have that all taken without any compensation, without any benefit sharing, you undermine the convention. And that's a major threat. For national governments, I think many national governments are now dealing with the reality that they've spent many years, or maybe they've only recently developed national access and benefit sharing laws, but in many cases they're built around this model of the material transfer agreement and physically sending cuttings of plants or whatever it would be from one country to another. So this is a major challenge for national governments. And national governments' sovereignty over their genetic resources, which is fundamental to the Biodiversity Convention, is also being threatened by this new digital world. You know, for indigenous people and local communities, they've always suffered from biopiracy. It's an ongoing problem. It's never been resolved. There's more consciousness of it. There are more efforts to prevent it now as a result of the Nagoya Protocol in particular and implementing these national laws. But this creates a whole nother avenue, and it's a whole nother way for biopiracy to take place, which is even less detectable than it was previously. And it also places national governments and IPLCs in a precarious position, in the sense that national governments have obligations to indigenous people and local communities under the Biodiversity Convention. But if the national governments are also being flummoxed, you know, by digital access, they might not be able to, you know, hold up their obligations. And finally, for sort of all of us, for civil society, I think that the way things stand now with these open access databases, which are effectively a free-for-all for sort of corporate patenting and pillage and conversion into proprietary products, is that the way things are going right now, it's sort of increasing corporate control over biodiversity and disempowering people. And I think that the solutions that will be generated through this paradigm of artificial intelligence and massive data are ones that will probably lead to further alienation between people and reality. You know, the sort of urban myth, maybe it's true, story of people that don't understand that milk comes from cows these days. Well, I think that problem will be exacerbated by the manner of understanding and relating to nature that the technological developments as they're going now point to. So that in effect is my presentation. I hope that you're eager for answers. I hope that you're interested in, well, so what are we going to do about this? How can we do this? How can we stop this? How can we make this more fair? We can start to talk about that in the presentations, or excuse me, in the Q&A. And if not, definitely attend the next webinar, because we will talk about that then. And I apologize for having gone a bit over, but do I go back to you, Bharat?
Thank you so much, Edward, for that wonderful presentation and explaining to us about the inner issues related to DSI and inequalities related to DSI. We hope to listen to you again at the end, and I hope that many will have many questions. Now, right now, moving, the next speaker is really very interesting. I am actually very inspired by her talks when I checked her speech in YouTube videos. It's really very interesting, and I could learn something. We have Ms. Anita Gurumurthy. Anita Gurumurthy is a founding member and executive director of IT for Change, where she leads research collaborations and projects in relation to the network society with a focus on governance, democracy, and gender justice. Her work reflects a keen interest in Southern frameworks and the political economy of internet governance and data and surveillance. Thank you so much, Anita, for joining. Now we look forward to listening to you.
Thank you so much. It's a pleasure to be here. I'll try to follow Bharat's suggestion and advice as to keep my pace slow. Just want to check if I'm audible, I'm clear. And Edward, are you able to project my slides? Yes, thanks. So I'm presuming I'm audible. So Edward has kind of set the bar very high. My slides are not so interesting, but I do think that in continuation of the issues that he has laid out, I'd like to open up a little bit more on issues of big data in relation to biodiversity. In fact, one of the slides that he showed us right at the end indicated to us that we are moving from a particular paradigm into another paradigm. And I would like to actually capture that movement as a movement, not as a movement that stops distinguishing between what may be nature and what may be nature captured in data. This is not to say that nature goes away. We will still need to get milk from mammals, but the fact is that the domain of biodiversity may be understood, interpreted, and construed through the reality of big data. So slide number 2. What I'd like to do is, before I get into the connections and the relationships between biodiversity and data, talk a little bit about the phenomenon of datafication. Next slide. It's not as if we didn't used to understand data or datum as that piece of information that pertained to a phenomenon represented through itself. So the idea of data always existed, and the idea that statistical information can give us a particular representation of reality has always been part of the realm of innovation, science, and technology. But what big data and related technologies in the form of machine learning have done is to really go into a realm of modeling and prediction, which has been unprecedented. So in some sense today, what we actually see is that datafication is extending to so many realms of our lived reality, which is that oftentimes when you're driving a car, which is a smart car, the car itself is a piece of digital artifact. It's actually something that is more digital than used to be an automobile as we understood it. Similarly, in different parts of the world, not so much in the developing or global south, we actually see that the refrigerator or the television set or any other artifact at home can actually be an extension of the ways in which we code our priorities. So you can set the refrigerator for a particular time, you can set the stove for a particular function, the television will talk back to you, you can speak to Alexa, and Netflix will identify priorities for you that Netflix thinks is a film or a TV serial you must watch. So in some sense, there is this pervasive interference, if you will, of a datafied society in everyday reality. And this, I think, is extremely important to understand, because the crystallization of our everyday behavior, the crystallization of information about our natural surroundings, the crystallization of information about lived environments like city life, for instance, commuting behavior of passengers that Uber captures. All of these are data sets that can talk to each other. What we need to remember is that one piece of data about you or me is actually not really valuable. It acquires a certain value when it is connected with other pieces of information. So it would be possible to look at a data set where patterns of behavior become particularly important. The interesting thing is that these patterns, discerned through masses and masses of data, lend themselves to serendipitous kinds of insights. What's important, therefore, for Netflix is to have volumes of information about yourself and people like yourself, and to be able to build prediction algorithm which can then nudge you towards watching what they think you should watch. So it is a large-scale behavioral project in the world. And this is extremely important also for the production and supply chains that is often referred to as the future of work. The future, I mean, this is an expression you may have heard, which is about how digital industrialization is going to replace the kinds of industrialization we saw at the turn of modernity. And digital industrialization is going to be distilling insights about all kinds of objects and organisms, connect them in very random ways and predictable ways to build models that can then become the future of supply chains. And this is an advantage that very, very few countries have. Next slide. This creep of data, or this creeping datafication, is also to be understood as something that is absolutely ungoverned. So what we have today is, if you set up a company like Amazon, and if you were Jeff Bezos, then you actually control massive amounts of information just owing to the fact that you got there first. So as an e-commerce company, you got there first, or as a travel app like Uber, you got there first, or in any other sense, any app that controls, let's say, the tourism industry or the streaming apps that we use, all of these are near monopolies. Why are they near monopolies? Because they took advantage of the internet, and through that network advantage, and because of what is called in economics as the first mover advantage, they managed to connect different users, gathered the data of these users, and then self-declared that they are the owners of this data. So in some sense, the data that flows across borders from different users to the corporations that seem to be controlling this data, that is part of a large governance deficit or a democratic deficit in the way in which data as a resource, or big data as a resource, is managed. So in some sense, the biorevolution that somehow gets dovetailed with the big data revolution is now facing a crisis, as Edward pointed out, because of the huge economic implications of data being able to flow across borders without any regulation. Next slide. This is captured very well in the words of Sean Ward, who says recently, as working with the physical world is becoming increasingly digital, every company that is out there is discovering that they are either a technology business or they are dead. And that is what is happening to biology. It is becoming a technology business. The next slide. What we need to see, and I think Edward did this in a very solid way. He showed us how from, coding one bacterium over zillions of years, in a short span of 2 decades, we've managed actually to do, what we have at scale, right? This kind of the spike in innovation is reflected in the fact that pharma or big pharma is today investing 5 times as much in research and development as it was 2 decades ago. And this investment, as my subsequent slides will show, is owing to the huge opportunity they see in DSI. Next slide. All the 10 so-called big pharma companies have either expressly collaborated with or acquired AI businesses, and they want to really take advantage of the benefits that artificial intelligence brings to the table. Next slide. In healthcare, I would like to point to some dramatic developments. Big tech has also been increasing its investment in healthcare, and in Jan 2021, Google acquired Fitbit. And this was no coincidence. With the lockdown, with COVID, people were sitting at home, and certain businesses, in a very, very perverse sense, as the world economy or the real economy was not doing very well, lots of e-commerce companies started doing better because our lifestyles changed dramatically. So Fitbit was acquired by Google because there's a huge opportunity for self-tracking, and people got into this kind of mode of exercising and quantifying themselves through these self-tracking wearables. Microsoft also announced its intent to acquire another communications company that does analytics in healthcare. Next slide. So if you really look at the ways in which this has grown over just 1 year, you can actually see the jump, right? In terms of how venture capital has gotten into the connections between technology and healthcare. And that the chart is self-explanatory, so I will leave it at that. Next slide. What we must watch out for is Google's investments in healthcare. So if you actually look at these companies, Apple, the GAFAM, as they are called, those big tech, the 5 big ones, Google is really leading the way. Next slide. What's of interest to us is big tech is also investing very heavily in genomics. And wellness and genomics are 2 very important industries. And just in a minute, I'll tell you why. Next slide. That's because a lot of this has to do with the marketization of that which was earlier not necessarily in the commercial sector. So with the diagnostics, of course, we have seen a certain move over the past 30 years with hardware and software coming together. But in agriculture and personalized medicine, especially, we're actually seeing a lot of competition, and big pharma and big tech are coming together to offer you products that you never knew you needed. So that is really the kind of frontier where reality is going to be in a certain way engineered through preferences that commercial interests will set for you. Now I go to the next set of slides that I want to talk about. Next slide. This is going to be the connections between data flows and DSI. So the next slide. And this is something that's a little bit difficult to understand, so I will go slow. And Edward, I hope we are on this slide. I'm just going to check. Yes, we are on the right slide. Why is it important to understand the data regime or the de facto data regime, which is not governed through any kind of international norm setting? It's important because even though there is no international law or there are no international standards for how data can flow, there are ways by which data is understood, as I already mentioned, to be the private property of certain corporations from predominantly the US, followed by China. In the absence of an overarching multilateral framework to govern this space, what happens is bilateral and plurilateral e-commerce agreements become the route through which the rules of the game are set. So if my country enters into a free trade agreement, let's say with the US, invariably, the US will actually have three main templatized cookie cutter rules in the free trade agreement. And this is like a copy paste. One, it will say, in these sectors where we agree to collaborate and where you agree to liberalize, you will allow data from that sector to flow freely to my country. So if it's agriculture, then all agricultural and farm data will flow freely. And within the context of the agreement, of course, it would be a mutual free flow. But we know that in reality, it is the companies from the US that are better placed in the domain of datafication than the companies that are located in the global south. So invariably, it is actually not a win-win situation. The second templatized rule is one that prohibits data localization. So the companies will say, or the free trade agreements will say, you cannot, as the government, force our companies to keep a copy of the data in your country. You have to compulsorily allow this data to flow without requiring the data to be housed in servers in your country. The third de facto or default rule that comes through the free trade agreement is immunity from source code disclosure, which means that if there is an algorithm that is being used by a foreign company in my country or in my national jurisdiction, the government of my country will not be able to open up that algorithm, even if it suspects that there is something hanky-panky about it, even if it suspects that Amazon is using an algorithm to distort the market. So what this means is that you're not only taking away the data, but I am unable to scrutinize the malpractices that you bring back in the form of your artificial intelligence-based algorithms. This is really important to understand because data is today being regulated by an instrument that it was not meant to be regulated by. Trade deals become the route through which data is regulated when actually data as a resource needs its own governance mechanisms. So this is very important to understand because when we talk about genomic data and the various efforts by the World Economic Forum, including the Earth Code project that Edward spoke about, or the Global Alliance for Genomics and Health, we are finding it a very big problem that we cannot ask many companies to localize this information that they are taking away. Next. This kind of laissez-faire, free for all, or finders, keepers logic puts DSI in the hands of big pharma and big tech in a way that we cannot really claim back this data, which creates what in economics is called a private enclosure. We cannot, or our governments cannot, our municipalities cannot, our village level councils cannot use this data that is being stolen. And as Ching said right at the beginning, this theft cannot be contained because we are unable to use the very same big data sets for the betterment of humanity, the betterment of communities and societies. And invariably, all of this tends to be locked in these meritocratic, reputational enclaves where researchers who want to publish these gene sequences seem to be wanting to get credit for it. They put it in the open access databases. The open access databases are watched by the big companies. The big companies take these dispersed codes, put them all together. They try to do a mix and match. Of course, I'm speaking exactly like a non-biologist, and so forgive me. I know I must be making scientific mistakes, but the point of the matter is that these databases easily allow what Edward so poignantly pointed out as the rebirth of an organism just through an email, right? So what we're actually seeing is profiteering through big data enclosures has become the de facto or the default paradigm in which we are functioning. Next. There are a couple of examples. I would just gloss over them because I would encourage you to read through these. And the Gen Bank has already been mentioned by Edward. And so you should look at the story of Ebola and how Regeneron profiteered from the sequence that was used from the Guinean sequence that was obtained from the country. Similarly, the GMO potato story is also very interesting, where the samples have been collected and reproduced in a lab in Peru, and this has been backed by the developed countries, the richer countries of the world, and certain very, very key philanthropic foundations at the cost of small farmers who produce local varieties. So I just wanted to flag this, and this is public data, mind you. What we need to understand, next slide, is all of this is possible not only because of the miracles of technology in terms of what can be done through data, but also because of the economics of this technology. Why is it that just for $300 you can get your sequence information as an individual? It's because the digital and data revolution have also broken that frontier, which was a cost frontier. If you put 2 and 2 together, that is my slides that I showed you before and this slide, ideally speaking, this kind of lowering of costs should enable developing countries also to benefit from genetic sequencing. But the fact that their data is stolen and their data is taken away and corporatized and privatized behind patent regimes is the reason why the economics of the data revolution has not really been able to create an ideal world. Next slide. So, what happened during the pandemic to DSI? One thing that I'd like to point to is what is now unfolding in the politics of developing and developed countries contesting the ways in which the WHO has set up the BioHub, which is meant for the rapid and broad sharing of pathogens for surveillance, research, and timely medical response. The WHO has also announced the setting up of the Biobank, which is for sharing or voluntary sharing of pathogen materials. And all of this is being presented through a moral imperative to share freely, right? That we all should share freely in this universe where this one particular virus is, wreaking havoc, causing mayhem. And therefore, the development of effective vaccines for humanity depends on all of us, all our countries, voluntarily pooling the data sets. But what we also know is this huge problem that the previous regime under the Nagoya Protocol would call for equitable sharing of the benefits, right? And that was the huge fight. And the biobank, unfortunately, will lead us to a situation where the benefits of this kind of free sharing are not necessarily going to accrue to the universe. Just the other day, the Director General of the WHO appealed to the conscience of the leaders of the developed world that please wait for the booster shots because less than 10% of the populations of developing countries have even got their first shot. So we know the paradoxes of this kind of, the DSI paradigm at this particular contemporary moment. 400, next slide. 400 civil society organizations last year pointed out that all of this information that we are giving should actually result not just in the sharing of pathogen information one way, but should result in a mutual kind of technology transfer where not just medicines, but everything from diagnostics to vaccines should be made available. Unfortunately, 1 year down the line, the reality or the writing is on the wall. The next slide. I'd like to speak a little bit about open washing. Just like pink washing is very popular these days in the advertising world, you want to sound very inclusive, you use these kind of moral tools to appeal, which is called something, right? It's called virtuous, virtue shopping or something like that. The word fails me. But basically, openness has become a kind of terminology which is very fashionable. It is nothing other than the reification of innovation for commercialization. You keep on talking to this kind of rhetorical, rhetorical hype about openness, which is nothing other than open washing. And I'd like to encourage you to see this wonderful report written by, authored by two people two experts in France, and it was endorsed by 100 experts. And it's a very, very sharp story of 2017 when France decided to return a lot of cultural heritage to Africa. But the report of this kind of restitution of the cultural heritage that the colonial power held, the recommendation of the official report said, let us do a systematic digitalization. Let us make sure that open access requirements are all, completely observed and intellectual property rights are fully respected during this restitution. So the experts said, if indeed the French government recognizes that African cultural heritage is not theirs to keep, it should not be theirs to digitize and make available in open access either. It seems very logical to us, but this is the contention that Edward is saying is going to be haunting us in the Biodiversity Convention, COP, that's going to come up later this year. Decolonization will never be achieved till we are able to govern digitalization and datafication also from the perspective of calling out the double standards of the developed world and their corporations in terms of the recolonization being attempted through datafication. Next slide. So finally, I didn't know that this is going to be a teaser and we are going to contemplate solutions in the next webinar. So maybe I'll just take much less time, maybe a minute. So next slide. I think we do need to remember that there is a stalemate today between the global north and the south that Edward spoke about in very crystal clear ways. Does the use of DSI qualify for sharing of benefits? Who is getting the benefits? What exactly constitutes the sharing of benefits? So is it monetary? Is it kindness of heart? Is it, data for good initiatives where big companies will say, I am willing to share, I'm Facebook, I am willing to share data about child pornography, which seems to have increased during these COVID months. So I will be more than happy to share this data with you. Should we allow companies to, through their benevolence, share data with us? That's a very important question. Also deeply related to biodiversity-based commercial and non-commercial research. What should we be doing about managing our resources, our natural resources, which are really under deep stress because COVID has really completely squelched the budgets of countries to be able to take care of its environment. And in the global south, the humanitarian crisis and the crisis of the environment, the crisis of climate are staring at us because of the post-COVID conditions. We would also have to contemplate how this non-monetary benefit sharing can ever be accomplished without a huge injustice through open access databases. And my next and last slide. I was just going to point to some solutions, but I would just leave you with this slide, just saying one or two things. I think it's really important to look at the connection between why biodiversity and intellectual property need to be connected. Why the regime of intellectual property has really wreaked havoc with the ways in which indigenous people or countries and communities of the global south have never ever been able to assert their rightful claims over their natural resources. This fight was fought even before the Nagoya Protocol, but somehow there was an uneasy truce, and it was always believed that the realm of resource governance has to be looked at from a Western lens, that you can never confuse cultural resources, social commons with the natural commons. You have to keep it aside. But not only for the indigenous people of the world, including the indigenous people in many parts of Latin America, Asia, and of course, some parts of Africa, the vital thing is that a communitarian approach, the Ubuntu approach, for instance, through which the free software movements have organized themselves, the ideas of the commons somehow bridges natural and cultural resources. So if you go beyond Western conceptions, we really have to address the idea that data governance has to really somehow take into account or take cognizance of the fact that unless you think of new governance frameworks situated within the management of the collective commons, you will never be able to find a suitable or appropriate solution that can guarantee that the big data paradigm can be rescued for the benefit of public interest, for the benefit of social value, right? Today, we're talking about private and commercial value, not so much public value and social value. Public value, a very important example in the climate context I can give is if only the energy management solutions of a city are not smartified. We don't need these smart companies to come and tell us what exactly we need to do with our private electrical equipment. But if our municipalities were able to put out transparent data and identified hubs where excess utilization of electricity happens, invariably the gentrified hubs of the city, right? We will never be able to have the transparency, the informational databases, informational transparency the public needs in order to be able to take action on how a city can manage its energy consumption, right? So these kinds of public value creation through data and social value creation for cultural, kind of communitarian ends, right? Local knowledge commons, exchanging ideas about how to go organic, for instance, grow a city garden. These are really in the realm of value creation through data that connects the natural world with the data world. And the caution I want to place here is that DSI is an exemplar of the way in which everything about this connection has gone wrong or is going wrong. I'll just stop there.
Thank you so much, Anita. I hope I'm audible. There was some network issue here. Okay, thank you so much, Anita, for sharing that wonderful, very enlightening and insightful information about data, what exactly is data and how data has been used to manipulate also in our behavioral aspects. Now, in one of your slides, actually, you pointed out whether DSI will have any benefits for like to the people who actually protect biodiversity. So I think I'm going to cover with some case studies so that we'll have some stories from the past, how DSI has been used. So I think the next speaker is myself. I hope I'm audible. I request the participants kindly interact with me during this session. You can use the chat box for your questions because I don't want to be this only on from one side. Yes, I think I'm going to share my few case studies, not much, to explain how DSI has been till now being used. Thank you so much, everyone, for joining. I am Bharat Reddy. I'm from Hyderabad, and I have worked as a technical expert advisor with Central Indian Biodiversity Board, and presently I'm working with Global Youth Biodiversity Network. So I'm going to share in this slide few case studies related to DSI. Now, before I share some about DSI, I just want to tell one story. We all love our mothers because she cooks really well, and especially if your mother is special, then she can make wonderful recipes for all of us. Now, because of that, one day you have invited a guest to your home for a lunch, and the guest came and he ate the food and he liked it so much. He asked your mother what are the ingredients and recipes that you have used, and you have no idea what's going on in the mind of that guest. Now the guest collected all the information, and the recipe of your mother is sold in the market at a price that you are actually not aware of. So this is actually something we can correlate to DSI, but how in the past some cases have been used. Before we get into, give me a second, I'm having some issues here with the internet. I hope you all can see my screen. Okay, thank you. Before we get into DSI, first of all, let's understand very simple about Nagoya Protocol, which came into force. A protocol, it's a supplementary protocol of CBD, which talks about fair and equitable sharing of benefits arising out of utilization of genetic resources, which is one of the objectives of CBD. Now, as you can see in the picture, what is the main purpose of Nagoya Protocol? That the benefits should be shared with the communities or with the origin country, which raised in the market. So this is very basic about Nagoya Protocol, but how it has been implemented, there are many challenges related to it. So right now, the entire debate about DSI is that whether it should be included under benefit sharing or not. That is the entire debate happening at the, under the CBD, under FAO, etc. Now, let us understand how CBD defines genetic resources. Well, there are two definitions under CBD instrument. One is defined by the CBD convention text itself, and the other is the Nagoya protocol. The CBD protocol defines genetic resource as genetic material of actual or potential value, and genetic material is defined as any material of plant, animal, microbial, or others as functional units of heredity. So it indirectly telling that both tangible and intangible forms of genetic resources are covered under this. Now, Nagoya Protocol went ahead and defines, like, in a very much better way, that and it talks about utilization of genetic resources under Nagoya Protocol. It reads as research and development on genetic and biochemical composition of genetic resources, including through the application of biotechnology. So, these are the two definitions under the CBD, about genetic resources. You can interpret in any way possible, because CBD is a general framework. It gives the rights to the parties to frame their own laws, but it should be very parallel to these definitions. Well, when Nagoya Protocol came, it was in 2014. Most of the people were there in Pyeongchang, and there was a lot of appreciation, and there was a lot of, great thing that Nagoya Protocol came, and we are going to conserve the biodiversity. Well, I think, for everything, once you think there is a solution, then there will be another problem. Then the problems keep coming. So let's see what are those things that I'm going to share with you all. Before I introduce you to these case studies, first of all, you should know about these terms which are used, which are material transfer agreement, a contract that governs the transfer of materials between institutions for use in research. The next term is mutually agreed terms. In short, we call MAT, an agreement reached between the provider of genetic resources and a user with respect to the conditions of access to genetic resources in the provided country and the benefits to be shared between both the parties for the commercial use of these resources. There will be a term called Ongole. Ongole is a town in eastern coast of South India. UNICLOS, I think most of you might be knowing, UN Convention on the Law of the Sea, and DSMZ, which is German Institute of Collection of Microorganisms and Cell Culture. So let us go to the case studies. Well, the first case study, ladies and gentlemen, is the future of Okavango. I think most of you know where is Okavango. Okavango is a delta in Botswana and also Namibia. So this case is related to Namibia. So this future of Okavango was a large interdisciplinary multi-institutional project. It was actually aimed at promoting sustainable resource use in the Okavango Delta in the face of climate change and land use conflicts. Now, what happened here was that it's a project between Namibian Institute and the DSMZ. The main purpose of this particular project was to study the interaction between bacteria, soil, and its role in soil fertility. Now, the project was all the scientists from the DSMZ and the Namibian Botanical Research Institute were combinedly studying the soil samples, etc. But one day, DSMZ thought of collecting soil samples for investigation. And by the way, this was before 2014, like before Nagoya Protocol was signed. Now, because Nagoya Protocol was not into force, both the DSMZ and Namibian government they have to sign something called as MTA, that is Material Transfer Agreement. Now, this Material Transfer Agreement was signed with many clauses in it. It talks, the clauses talks about limited to non-commercial research, benefit sharing should be shared, and commercialization of the material is prohibited. And to do all these things, they have to take permission from the Namibian authorities, etc., etc. Now, what happened here is that these particular soil samples, they collected the soil samples to the Germany and they started to culture it. And later on, they started to identify the bacteria that are present, and it was happening in a very non-commercial way. So, I mean, in the beginning, all the soil samples were identified, and the bacteria got like extracted, and the taxonomic identification was given, but genomic sequence was still not done. Remember this point. It was only being done to identify the bacteria and all those things. Of course, even to identify bacteria, because it's intangible, you have to do genome sequencing, but it was not kind of published yet. Now, later on, what happened was that because new bacteria were identified, this is very important. Because new bacteria got identified, now there is a bacterial code. If you all go through this bacterial code is related to international nomenclature for prokaryotes, it says that once any new bacteria is identified, the microbes should be deposited in 2 separate culture collections in 2 different countries. Now, this is the clause related to bacterial code. So, Belgium, actually that time Belgium thought like, they can actually collect these bacteria samples and they can actually store them. But even to do that, the German, the DSMZ has to approach the Namibian authorities to take permission. Now they took the permission got like, they gave the permission, and this time there was another MTA. This is second MTA, Material Transfer Agreement 2. Now, what is this Material Transfer Agreement 2? It's very similar to the first Material Transfer Agreement, but here there is one interesting thing they have added this time. Look, it says that use for commercial purpose, including genomic sequences or any use for commercial application, is excluded and requires prior written consent from the Namibian Botanical Research Institute. This was the clause they have added. Now, I think the samples were only collected and the bacteria were like cultured, but they were not yet published. So till now, this is the half story of this case. Now, what I'm going to share is the second half story. Now, this is very interesting because Nagoya Protocol came into force in the year 2014. And by this time, Namibian government already have their ABS guidelines and etc. So in 2016, to further study and to extend the project of future of Okavango, the DSMZ obtained permission this time for full genomic sequencing from the national government of Namibia based on a permit. Now this particular thing they have done so that they won't violate the clause 9 that I have shared in my previous slide. So they kind of took the permission, and they took the permission for full genomic sequencing. Now, again, the Namibian government were like super conscious about this thing, that they are asking for full genomic sequencing. So what shall we do? So I think they developed a new guidelines this time. So it was very similar. No, I mean, it talks about no benefit sharing, and commercial research is not allowed, and they have to do all the things. But there was no text. There was no text that says that permission is required for the publication of genomic sequence. So this was the major thing that that was not added in the in the guide in the agreement itself. So the bacterial strains now presently completely under the control of DSMZ. The Namibian authority, of course, DSMZ being so, like, they include all the information related to from where the bacteria they have identified. They give credits to the Namibian authorities. All the material transfer agreements are actually put in the online site, and people who wants to buy, companies who wants to buy, they can do it. Because you see, there is no wording called publication of genomic sequences. So the wording publication is very important. Now, one other interesting thing is, even if Namibian authorities wants to add They can't. Why? Because Germany falls under European Union. Now, the European regulation about ABS is very interesting. In order to fall under the compliance measure of the EU regulation, genetic resources must have been accessed in situ or on or after 12th October 2014 and must be collected from a party to the Nagoya Protocol, in which ABS legislative or regulatory requirements apply. Thus, the bacteria covered by the MTAs in the Okawongo case actually fall outside the scope of the EU regulation. So EU can't do anything to DSMZ. They can't direct them, hey, you have collected illegally. I mean, you cannot control in your like online website about this. You cannot like, publish the genomic sequencing of this data. So this entire case is about wordings, where Namibian authorities may not have put all the wordings in proper, and they forgot to mention something related to publications. So there was different interpretations about this thing. So what is that we can learn from this is wordings are very, very important if you want to sign, if you want to do material transfer agreement and mutual agreed terms. And these two are generally applicable when there is a bilateral mechanism, means between the providers and users of the genetic resources. So wordings are very important, and contradictory interpretations are quite possible. And you never know when once you publish, once from the DSMZ website, anyone can actually take these publications, and you never know the third parties. Who are these third parties? It can be done. So what I mean to say is the entire bacteria that actually got extracted from Namibia are under the control of DSMZ. There is a good collaboration, by the way. It's not like very bad. It's not a very complicated case, but it's a case to be considered. So I think this is one case that is related to material transfer agreement. So I hope that if it's technical, I know it's a little bit of technical, but I hope you could understand a bit. The second case that I am going to talk is between Oxford University and India. Now, Plasmodium vivax sample, Oxford University discovered new strains of Plasmodium vivax, and they applied for patent. And these Plasmodium vivax strains originated from different countries, including India. Now, what happened was, in this case, the Plasmodium sample strains, they collected from Indian patients, and Oxford University in the patent application wrote about this information that, yeah, we have collected the strains from the Indian patients, and they have mentioned it. And they send it to the patent authority of India, that is the Indian Patent Office, to gain the patency. But, NBA, National Biodiversity Authority of India, so we have we have the national law. So they acted looking at this case. Now, how they acted by using this particular, in the section C, it has two terms, namely traditional knowledge and associated knowledge. If you look at the law of India, then there are two terms. The traditional knowledge, as you might know what it means, but associated knowledge, they have mentioned very specifically that according to access on benefit sharing guidelines, because India is part of Nagoya, the term associated knowledge is scientific knowledge. So DSI is under obligation of access on benefit sharing. So NBA kind of immediately sent notice to Oxford University, the institute that develops vaccines, saying that, hey, you have to stop immediately this vaccine patent. It's not yet comes under the invention. Unless and until you take permission from us, you can't file the petition. You can't file the patent. But what did Oxford University did? They did remarkably, amazingly. The Oxford University deleted all references to the Indian strain from the application. And they claimed that the nucleotide sequence of this strain was obtained from a 2005 journal publication. This is like amazing thing that they did, but the Indian government didn't give up. What they did, they actually went ahead and checked the applications actually Oxford filed in various jurisdictions. Now, Oxford being so Oxford, they forgot to delete or amend the same point in other applications. I don't know what to call it. Should be comedy of errors. So now, because they have forgotten to delete all these references in other jurisdictions where they have filed for patency, the applicant continues to be in the violation of the requirement of Biodiversity Act in respect of obtaining patent. So what is the two learnings that we can get from these two cases is that I was reading something from International Chamber of Commerce where they say that intangible genetic resources are not part of CBD. CBD doesn't cover. If these are not intangible, then what it is? So I hope that these two case studies, I just wanted to explain how DSI or anything related to DSI have been done in the past. Well, I think the next is very similar to not very close to DSI. I mean, it's not completely DSI, I would say, but can be DSI in the future. Now, this amazing bull, I have good association with this bull from my grandparents. It's called Ongole bull. As I mentioned, Ongole is a place in the east coastal side of east coastal region of South India. The bull is very massive. The bull is economic value for the farmers. Now, this bull got smuggled during 1950s by Brazilian firms. And I think that time the amount could be somewhere around maybe $100 or something. And presently this Ongole bull is sold. I mean, a decade ago or 2 decades ago, this Ongole bull used to get sold around somewhere around, $2,000 to $3,000. But Brazilian firms, they take this Ongole bull and they did cattle breeding with the local breeds, and they developed the breed varieties of Ongole bull. All the Mexican bullfights, all the Latin American bullfights you guys see in the television, thanks to this Ongole bull. Because of this Ongole bull, of this original breed, your Latin American breeds got developed. Now the demand for Ongole bull in Latin America is more than $200,000. Can you believe that? So a bull which has been culturally, spiritually, economically very important to its origin country. People connect with Ongole bull. They pray to Ongole bull. Is valued economically more than its own country. Now, I think I tell you why the reason, we don't take care of this kind of things. And by the way, more Ongole bulls are there in Latin America than India. So, I mean, this is very interesting. I was actually looking. This is a funny quote I got from one of the bureaucrats. When you have office or bureaucrats like this, can you expect that biodiversity should be conserved? Now, what he says, very interestingly, during my visit to Brazil, I discovered that they had improved the Ongole bull variety genetically. There are more than 1 crore top quality Ongole bulls in Brazil and Mexico. According to me, there are not more than 1,000 Ongole bull top breeds in India. This is really, when you have authorities like this, bureaucrats like this, how can you expect, ladies and gentlemen, to conserve biodiversity and sustainable management? So somewhere we are lacking that in that aspect. Now Brazil is keep coming to, I don't, when I say Brazil, it's the Brazilian firms. And I want to tell one more interesting thing here. Even the Indian like foreign diplomacy and also the Brazilian diplomacy, especially the Indian diplomacy, they don't want to act on this because it's a great economic value to Brazil, and India and Brazil have great connections. I mean, in terms of trade and everything, because we are part of BRICS. So Brazil keep on saving the Ongol bull. Brazil is keen on saving the Ongol bull right now. They are telling they want to transfer the technology to India and everything. So somewhere I think Brazilian firms who are responsible for this, I think India owes an apology from Brazilian firms. Why I'm saying? Because of this. The farmers who have been protecting Ongol bulls for thousands of years, because of one drought, made them to sell the bull less than $500. If it is not touching, if it is not emotional to you, then you are not in the right field. No authorities actually took care of them. Imagine the farmers who have been conserving, if they have realized their potential of Ongol bull, economically, they have been well settled. Why we are talking about post 2020? If you can't implement anything at the grassroots level. 2010, when Aichi targets came, but these farmers were selling the Ongol bulls, a pure breed, in a local market, just for their own survival. And by the way, many of them committed suicide. Are you aware of these cases? I'm asking the Brazilian firms. That's my question. Well, till now I have shared everything related to terrestrial, but if you think DSI is only related, is only like happening on the terrestrial land, no, it's also happening on the sea. Now, the whale that I'm sharing here, it's called sperm whale. It has this great, chemical called as ambergris. It's known as whale vomit. So these are like residues of the, the fecal matters of the sperm whale. So anyone who goes to ocean, generally they kind of collect this, and there is huge value for this. I think it earns in like $1 million. The business is in $1 million. Why? Because this ambergris produces a chemical which is related to very close to cholesterol, called as ambergris, and it has a very powerful, fragrant perfume smell. So this is used in perfume industry. I think the industry, if you know about this brand Gucci, they use the same chemical. I mean, actually they don't tell, but it's believed that they use it. So there are like, many like perfume industries where they use this. Now imagine if there is no control on the seas where, there is no jurisdiction of the countries, what can happen? It can be like, already there is a lot of big smuggling happens with this sperm whale, and many of the sperm whales get hunted for this ambergris. So in the future, I think maybe someone might be interested to do DSI, sequence it and sell it in the market. We never know. But this is an interesting case I was reading. I thought to share with you. Well, these are some case studies. I hope I didn't bore you, but I was attending the policy options as a representative from Gibbon, and there are 5 policy options right now, because I told you the debate entirely is about whether DSI should be part of CDD, access on benefit sharing. Now, the first policy criteria actually is whether DSI can be fully integrated into CBD and Nagoya Protocol. Means there will be like benefit sharing and everything will be there. The second is standard MAT. MAT is mutually agreed terms. It's a bilateral mechanism, and there is no restriction on access, but obligation on profit sharing. So it has again like two important terms, the second criteria use. One is bilateral mechanism, second is the multilateral mechanism. Now, the third option is multilateral fund, a big fund, multilateral international fund, a payment or contribution to go into the multilateral fund. One way is to like have membership, like people who wants to collect data, databases of DSI, they have to do a membership fee, and this money will go to the multilateral fund. And the other is a levy, a tax kind of thing imposed on products or services associated with DSI. And they are also talking about biodiversity bonds that have been proposed to this fund. The fourth one is very interesting. The countries that already have this technology, they go to the least developed countries or developing countries, to enhance the scientific capacity, and so that every country can have their own DSI policies, etc. The fifth policy, I think you all can read it. I don't want to like talk about it. Well, ladies and gentlemen, I think I am just showing this screen because, as Edward mentioned, that he don't have any solutions. I am very like young. I don't know. I also don't have any solutions. I'm learning about DSI. In my capacity, I'm sharing all the presentations, but I have certain qualifications related to DSI because I'm one of the person who did DNA synthesis in the labs and also did sequencing too. It has great potential that Edward shared, like identification of species and other things. Yeah, if you look at the pros, well, I think some of the worst things imaginable have been done with the best intentions. Now, as you can see on the left side of your screen, the entire biodiversity. On the right side, you can only see the sequences. Now you decide how you want to see biodiversity in the future. Now, I just want to share a couple of interesting things related to the best, the quote. When internet was actually started, someone asked the CEO, the founder of Microsoft, Bill Gates, about the possible consequences and threats related to internet. Bill Gates was paused for a second and said that nothing. His answer was nothing. But do you think we are not facing the problems related to hacking today? In the same way, Facebook was actually, the intention was to connect people, but today we are facing trollers everywhere. All the Microsoft, Google, all the top companies are going to artificial intelligence. Why? Because in the past there was protest from the employees, from the laborers. They don't want to do any, they don't want to like go into this protest and all. So they are going to artificial intelligence, but still they are not aware what consequences artificial intelligence can bring. In the same way, digital sequence information. You are sharing vast amount of data so aggressively, and you have no idea who is accessing that data on the other side. Even you can there are many third parties involved. You are not sure about hacking. You have not done anything on this. You are talking about blockchain technology, where, like in many countries, there are no proper laws on data. I think around 20 to 25% of the countries don't have proper laws on data. So how you are going to actually manage all these things? And I just want to tell, there is a community called as Do It Yourself community. Now, Do It Yourself community is a freelancer community where, as a biotechnologist, if I have knowledge on genetics, I can work at my own home. I can create a lab, and I can, keep inventing, keep manipulating, use all the gene editing, CRISPR, gene sequencing, etc., to create something innovative related to, any like new thing. That depends on me completely. If I have good intentions, I care for the society, then I think I use it for good. But if I don't have, then the consequences will be very, very, we can't even expect. For example, bioweapons. We are not sure about bioweapons yet. Now, in many countries, there are legal challenges. I think already Edward mentioned about this thing. there is no proper consensus on the definition of DSI. Even from the countries who support, they are very confused. Some countries are supporting or not. It's all like happening. So I think I leave these questions to all of you, and at the end, I recommend all of you to understand, if you want to understand genetics, if you want to like have a feel of genetics, then kindly watch these movies. Then I think it will like you will learn something. But at the end, I want to say that have compassion and believe in transformation. Thank you so much. Thank you so much for listening to me. I now invite Shruti to moderate the session on the Q&A. And dear participants, if you have any questions, kindly raise your hand and wait for the moderator to announce your name. Shruti, you have to unmute yourself.
Thank you, Bharat. Bharat, I would request you to, if you can give me some co-host this, because if we are expecting the participants to unmute themselves, we have to change the settings so that participants can also unmute and ask. Before we move on to the participants, I would check, because we've crossed our time limit by quite a bit. So is it okay with the participants if we extend for another half an hour? You could give me a thumbs up or unmute yourself and say yes. Perfect. Amazing. So we did have a bunch of questions that's come up. I would first take the list of questions that have come in the chat box, and then I would come to the raise hands, if that is okay with everyone. When I call upon the person may unmute, switch on the video whenever possible, and address who they want to ask the question to, and be as brief as possible, because we have a lot of questions to run through, and we have to finish it in half an hour. So starting with, do we have Andrea Morales? Andrea? Okay, we'll come back to Andrea's question. Do we have Verena?
Yes, we do.
Yes, please go ahead.
Okay, my first question goes to, oh my God, just a second, let me localize the question I need. Well, actually, I wanted to point something to the first presentation that I saw with a professor that talking about the germplasm bank. And I think it would be a very interesting application in the construction of those genetical banks, when we have to endure huge demand in the area of forest management, because we are having lots of advents with wildfires that are happening right now all around the world. We have cases in California, we have cases in Turkey and in Greece, also Australia in the last year. So I think it would be a very nice application of those technologies and those strategies to overcome those challenges that we have been facing on. And the second question I have is for Anita. Well, we have, actually, it's a fact that in this pandemics, lots of countries are seeking thriving, economical thriving. So we will have some sort of concentration of data, about genetical data to profit over it. So I would like to know about her, what she thinks, what's the tendency of those things happening, about countries concentrating informations about some specific organisms, such as virus or bacteria or fungi, or even some data about species. I would like to know a little bit about that. Thank you.
Maybe Edward and then Anita first. I mean, Anita next. So.
Okay, well, okay. So speaking about banks, right? There are longstanding issues in agriculture and in the Biodiversity Convention around the management of collections of biodiversity, generally referring to physical collections. When it comes to questions about sort of restoration of biodiversity that relies, when you have a disastrous situation and you've lost it in the field, that relies on physical samples of biodiversity. We're not nearly at the point where we would use sequence information to literally generate species. I think that the general hope and direction of the Biodiversity Convention is to preserve that stuff in what we say in situ, not removed from its natural environment, but rather growing in its natural environment. And for example, in agriculture, when you remove varieties of plants and put them into a seed bank, they cease to evolve and they cease to diversify. They become sort of a static, lump. So there's a role for ex situ collections. Unfortunately, one of them right now is that ex situ collections in Europe, in Belgium, I'm thinking of in particular, are becoming sources of DSI in quote unquote open access databases of African genetic resources, of Latin American genetic resources, of Asian genetic resources. So one of the problems that we have with these banks right now is that what are referred to as ex situ collections, so botanical gardens, gene banks, have in many ways are collections that were acquired in a colonial setting, in the colonial era, or in a sort of post-colonial era. And then now these things are being converted into DSI. I think Anita's French example goes to this question. These things are actually being converted into DSI and placed in open access databases, even though they really don't belong to the Belgian botanical garden or the British botanical garden or the Hawaiian botanical garden, whatever it may be. Just, I guess, one other related thought that I would have, and I'm not a professor, by the way, I'm just an activist like the rest of us. But the one other thought I would have would be there are a lot of interesting questions in the area of, we talked a little bit about pathogens and disease. There are a lot of interesting questions right now around the data banks of the sequence information of pathogens. And I think that there are reasons why. There's actually an agreement under the World Health Organization that provides for some real benefit sharing for influenza viruses, and so there's a question now, well, wow, maybe we could take a similar approach and apply it to other pathogens, because, as Anita said, it requires global surveillance and everybody around the world cooperating to identify these new strains, to sequence what's circulating. And without global cooperation, we don't have treatments, but the treatments are distributed very, very unfairly. So there's a lot of questions and a lot of movement right now. It's the kind of thing maybe to talk about in the next webinar. Well, what can we do if the whole world is coming together to bring together the digital sequence information that enables the creation of treatments? Why can't we get the treatments to the rest of the world? And maybe putting rules and terms and conditions around use of this sequence information is something that can be done to improve the world's access to these treatments, which are so unfairly distributed right now.
I think Edward has answered, what you wanted to understand about what's really happening in the post-COVID context, because I'm not an expert in that area. And what I'd say is that the current regime in its, is a one-way street. And that's really not on. But I also think that the crisis is really, really very big. And as activists, we really need to set the terms of the negotiation.
Thank you, Anita. Thank you, Edward. Please, I also would like to apologize in advance if I didn't get your name right, if I didn't pronounce your name correctly. We have Rui Obispo Guzman. Do we have? I think it was asked in Spanish. I can see Rui. I'm sorry, I'm not. Could one of the translators then ask the question if they're not able to? Could I request one of the translators to ask the same question?
I just wanted to say that there's one hand that is up, and I can see Christine's hand, just in case.
Yes, Anita. So I was just going through the list of people who have asked in the chat box. So the next question is from Patrice, and then I'll come to Christine, and then we have Anala. So that's the, so if not Rui, I would like to go to Patrice. Is Patrice around? Okay, so we have Christine. Christine, over to you. You can unmute yourself and ask.
Yeah. I have one compliment to say to Bharat about good intentions are no guarantee. But I want to point out that good intentions are no guarantee either in large corporations, but also not in small innovative teams. We've seen these co-opted youngsters at the COP in Cancun who will be serfs, even if they create small corporations, small companies, serves to the large corporations who give them prices and money. So it's not that easy that the large ones, they clearly need regulation because power needs regulation, but also the small ones are not automatically out of the good intentions are no guarantee. So simply want to congratulate Bharat on that sentence. Secondly, I have a question to Ed Hammond. I've looked closely into the commercial collections, and there is one commercial collection that I hope he will look into and do some research on. It's called Brain, B.R.I. BRIN, but with a dot after each one. It can be Googled easily. And they serve BASF, Bayer, DSM, Evonik, Henkel, Nutrinova, RWE, Sandoz, and others more. And what do they serve them with? They serve them with microorganisms, metagenomes, enzymes, whole metabolic pathways, biomolecules, and all the associated literature and cell collections and DSI, and they give advice on intellectual property right. Now, what happens is that nobody is going to get anything that makes sense if they go to a big data bank and say, give me everything that is good for sustainable development and human health. That's nonsense. You have to focus the question. And these commercial collections do the focusing, meaning give me all the plants that and fungi that have are known to reduce high blood pressure and associated with traditional knowledge, by the way. And then they look at them, see whether they have similarities in the sequences, and then comes the role of the big open data. If they have identified certain sequences, they go with that question to the open source big collections and say, please find me everything that has it in it. And that's far away. It may be soil organisms or something. And then they totally hide the traditional knowledge and the plants from what they got their focusing question and say, it's open source and we found it, and basically it's on this on the lawn, in the soil of the lawn of our administrative building. So these commercial collections play a crucial role to know which questions to ask the big open source data banks. And I would greatly appreciate if they were looked into more closely and their connection to the big data banks, because they access the big data banks and get from them what they need in the context of the patent applications of the companies they serve. And very often it's not the company name. They are not collecting in the company name. Brain is a middle-size commercial thing that even got environmental awards and has about 500, a team of 500. And so I think it would be good also to look at how do they find the focus? And this is where traditional knowledge comes in. And traditional knowledge, fortunately, is under the human rights regime, meaning it reaches back in time. So you don't have the time limits. And so I think in practical terms for regulation, it is very important to look at the role of traditional knowledge and also to look at the role of these commercial collections, which are not discussed at all. And we know very little about them. And this interaction between some of them with different names and the large corporations are a way to find out more about them. Thank you. So this is a task for Ed Hammond. I hope he's going to take it up.
Well, the expression that would be used here would be that you're preaching to the choir, Christine. I agree entirely with your sentiment. I think in the presentation, just to point out two things, some of the sort of logos and small images that I picked, I did for deliberate reasons. I put up the bit on Novozymes to try to point out, as one of the things you just don't have a chance to say everything in a presentation, how the enzyme and microbe business is a big one associated with this, and that people are generally unaware that their clothing, that lots of products that they buy that are not food, but then also a lot of food products are all the result of microbial processes that can be proprietary. And I think that in addition to what you said, the sort of private sort of gene bank type assistance or contractors to the big companies, I think that there's also another class, and they're exemplified to me by a company that's called Benson Hill in the US, which is an artificial intelligence provider basically to the agriculture industry. And they have what is basically, you know, a bloodhound dog that goes into these databases and tries to identify the valuable stuff. And so they lease out, and, you know, their artificial intelligence to the different companies to try to ferret out the specific information. And as you point out, and I think that all of us know that this must occur, but it's hard to place your finger on where it occurs. You know, if you find a sequence in one place and you identify its value, you might be able to find that same sequence in a more convenient place from a regulatory perspective and claim that you found it there and avoid all of this difficulty about benefit sharing from the corporate perspective. So I guess those are my observations. I would, you know, it's very frustrating having been working on DSI, you know, in the context of agriculture at the Food and Agriculture Organization, and in health, and at the Biodiversity Convention for a number of years. I can, particularly in agriculture and now in the CBD, there have been specific decisions that specifically mandated studies to be made on the extent and breadth of corporate DSI holdings, and reports always come back, we didn't find anything because it's impossible to know, because this is all private property, and the corporations did not give us access to their holdings. So it is an extremely frustrating situation. And what we do know, though, is that they fully leverage everything that they can that's public and supplement with what must be valuable information that we never find out even exists. So it is terribly frustrating, and I'll do my best with the resources available to me to try to advance that type of study.
I will give you some hints, and we totally agree that DSI is the perfect hiding place for very old-fashioned biopiracy.
Thank you, Christy. So there have been a whole bunch of questions for the speakers, Edward, Anita, Bharat, which I have copy-pasted on the chat box below. I think we are way past unless one of you want to go through the questions that I've copy-pasted here and want to respond to any of them. We could have the final round, and maybe with the permission of the speakers, if you could share your contact details on the chat box so that the participants can get in touch with you with more follow-up questions, if there are any, and take it offline, if that's okay with all of you. So maybe you can go through the questions that I've copy pasted in the chat box and may want to respond. So I can go with, you can go with Edward first, Anita, and then Bharat, if that's okay.
Sure. There are a couple of questions in the chat box that I would that I'd like to that I would like to approach. And this gets a bit into the meat of what I hope would be our next webinar, but it's good to talk about now because the questions are good ones. I think that you know, right now, what is under the Nagoya Protocol, what is universally recognized, well, yeah, effectively universally recognized, I haven't heard anyone disagree with this, is that for new access to biodiversity, so when, you know, when an exchange happens, you know, newly, you know, developing countries, provider countries are within their rights to put down rules about the sequence. They say, well, you can't sequence this, or if you sequence this, you have to give the sequence to us and we'll decide what we're going to do with it. All of these things, any approach in a bilateral sort of agreement is available to provider countries. Now, these sort of things, and in fact, in the Biodiversity Convention right now, there's an effort to create a global solution to this, maybe a multilateral solution to this. And this is a little bit of a maybe a threat that developing countries can make, say, well, if you're going to continue to just take all of our sequences and throw them in this database, which makes us unable to regulate, unable to get benefit sharing from them, well, we're just not going to let you sequence the material to begin with. Now, that is practically difficult to do, but it can be done. But I think that most people realize that that's not an ideal solution to this problem. You know, you don't want, and of course it opens up you to being accused of being anti-science or against, you know, technological progress, or in the area of health. They would say, you know, you're endangering global health if you try to prevent these things from being sequenced. So it's kind of so the question's a good one. You know, what is the legal framework? The answer is that we need to create it. And though there is that legal fallback that developing countries have to just say no. But again, it's practically kind of hard to do. You know, and so on the other question, I mean, you know, from the perspective of all of the developing countries, and certainly, you know, myself, if you use sequence information to access a genetic resource as a way to avoid benefit sharing, which I believe the US company Regeneron in the Ebola example did, you know, then from my perspective, that clearly is a violation of protocol. You know, so, but, you know, Japan, for example, will still maintain as a sovereign country in a negotiation that sequence information has nothing to do with the Biodiversity Convention. So we have to overcome these political hurdles to get to the point of getting there. And you know, and maybe that's a good place to stop, to say that there are a lot of people working on this, and there are a lot of challenges to developing the solution, but there's also reason to be hopeful that we can do it, and it involves, you know, an alliance of civil society, indigenous people, local communities, and the developing countries that are advancing the agenda to create benefit sharing for this stuff. And let's keep talking about it, and I hope to continue to talk about it. So thank you, and I'm signing off. So I'll share my email in the chat, and thank you to everyone who's been with us all the way to a good half hour beyond our scheduled end. Thanks. Bye bye.
Thank you, Edward. So I think we're gonna close the session. Before that, I'll just give it to Bharat to say a final vote of thanks. Bharat, over to you.
Thank you, Shruti. Thank you so much. Thank you, everyone, for joining. Thank you, Edward. Thank you, Anita. And thank you, everyone, for taking your time and joining and listening to this session. So with that, we will close our session on DSI. I hope it is helpful to everyone. And hopefully we'll have another session maybe in future about the policy levels at DSI. So I thank one more time everyone for joining. You all have a great day. Take care.